Well, hey guys, good morning. Welcome to the review for Test 2. It is now week 5 in the semester. We are past the halfway mark. We've learned a lot, and now it's time for Test 2. In this short video, I want to go over the test, the key concepts that will be on the test, and kind of help you prepare well. So the first question on the test is going to be kind of related to this idea of irreducible complexity. One of the things we've seen so far in the content we've learned is that while biology is simple, at least in some ways, it's actually quite complex, right? Even a simple single-celled organism is incredibly complex. There's so many things that are precise and fine-tuned and methodical. And so in question 1, we're going to spend a little time. I want you to be able to define the term irreducible complexity and explain the gist of that argument, how that is used by some as an argument, and I think a compelling argument, for the limits of evolution and the need for God, right? The need for an intelligent creator. The second test question will be multi-steps, and it's going to be a case that kind of walks you through tying in everything we talked about with the cell cycle. So you need to be able to explain the eukaryotic cell cycle as it relates to mitotic cell division, how checkpoints work in that process, and then how mutations in checkpoint proteins oftentimes leads to cancer, and kind of how that cancer biology works. And then combined with that, be able to use our genetics understanding. So we practice using Punnett squares to predict simple traits that are passed on that are autosomal dominant or recessive, where we got 2 alleles, and then be able to read a pedigree. So that's a big one. We're going to walk through that. And then question 3 is kind of tying in the molecular biology piece. So one of the central dogmas, the central themes to biology is the flow of information, right? From DNA to RNA to proteins. So we're going to kind of go through that. All right, so let's tackle the first one. All right, so question 1, I want you to be able to define irreducible complexity, explain how it's a good argument for the limitations of evolution and the need for a creator, and then think of one example that we learned in this unit and kind of break that down in terms of how that can be used as an example. So what is irreducible complexity? Simply put, it's the argument that some mechanisms in biology are so complex and all the parts are so interdependent that if one part was missing, the whole thing ceases to work, right? So the idea with natural selection is that there's genetic diversity. There may even be mutations that happen sometimes that give a new trait, an advantageous trait, right? And the idea with natural selection that drives evolution, we'll talk about this more next week, is that in a population of organisms of the same species, when there's a change in the environment, there is natural pressure, right? Limitation of food sources, pressures of competition, a new predator, whatever the case might be, changes in climate. That's going to put pressure on the population, meaning it's going to be harder for some to survive, reproduce, and propagate the species. So natural selection would kind of naturally select for a trait that is advantageous, right? So over time, as the environment puts pressure on particular traits, those traits become more prominent in the gene pool, and so we kind of get a change in the characteristics of a population over time. In the case of Darwinian evolution, or simply put, evolutionary theory, common descent, right? So extrapolating the phenomenon of evolution all the way back into history, right? Combined with this naturalistic philosophy that we've been kind of pinpointing in modern science, gives us this idea that all things, happen, all living organisms spontaneously arose from non-living things at some point and evolved from a single common ancestor, right? That's the story that you hear. And irreducible complexity is used as an argument for that. So there are some things, and Michael Behe, you can read more about him. I posted a YouTube video in Blackboard, but he's the one who kind of coined that phrase. And one of the examples he likes to use in his book called Darwin's Black Box is the flagellar motor, right? So in bacteria that are single-cell organisms, some move through a flagellum, right? this long whip-like extension from the cell that's driven by cytoskeletal proteins. And he breaks that down and says that all the parts are so necessary that if one is missing, the rotor doesn't work, right? You have no flagellum. There's no motion. And so he uses as an argument to say that if it's inconceivable that some mechanisms like the flagellum motor that are irreducibly complex could have arisen through unguided natural selection processes, right? Natural selection preserves traits that are advantageous. And so the idea then would be that organisms gradually evolve by saving traits and adding traits through mutations and natural selection that are advantageous, help them reproduce and make offspring. But irreducibly complex processes serve as a problem for that, right? There's not a clear mechanism for how these random, irrelevant pieces that have no function, that give no survivorship advantage, would be kind of hung on to until we can put all the pieces together, right? It just doesn't make sense. So that's kind of the idea for that argument. Some examples, there are many, right? We talked about the flow of information, how the genetic code is driving the storage and transfer of information, right? The fact that DNA is protected, particularly in eukaryotes, in the nucleus, and genes are uncoiled so that they can be precisely copied into mRNA, and then the mRNA gets processed just so that we get a functional message RNA, and then it carefully leaves the nucleus and encounters a ribosome, and then that ribosome reads the code and takes the corresponding tRNA molecules that have the amino acids and builds the protein chain, and then the protein unfolds. It's just a complex process, right? It's kind of hard to conceive of how that could spontaneously arise, right? If we conceive of the argument that simple proteins could have happened on their own, just through catalyzed reactions in the primitive ocean of Earth. How did we get to the process where not only were proteins replicated, but that was encoded in DNA, right, and preserved? It's just a good example. And you can think of others like that, right? The cell cycle, etc. Okay, so that's question one. Question two, it's a big one. There's a lot in this one. So this is going to be a multi-step question where you work through kind of a case. But the gist of this question is we want to be able to apply what we've learned about cell division, right, and genetics. So cell division is the process of a single cell multiplying itself. In the case of prokaryotes, bacteria, that's a simple process, just called binary fission, right? There are no organelles to reproduce. There is no nuclear membrane. The chromosomes are simple and smaller. So bacteria grow and reproduce and even evolve quickly. Eukaryotes, though, which are most other living organisms, plants, animals, fungi, protists, they are more complex, and oftentimes they are multicellular organisms. So we get eukaryotic cells that have a true nucleus, complex, large genomes and chromosomes, and so we get this complex process. So that is mitosis. So mitosis is the process of cell division in eukaryotes, and this particular type of cell division, mitosis, leads to genetically identical daughter cells, right? Meaning this cell that enters mitosis goes through this complex series of processes, and the end result is a clone of itself, right? We have two cells from the one, and they are identical. And the purpose of mitosis is growth and repair, right? So as organisms are growing and repairing, particularly multicellular organisms, that's the purpose of mitosis, is to sustain those multicellular organisms by replacing damaged cells. So in humans, we can think about, even an adult, we're making new skin layers as the more superficial layers die and slough off. Same with the lining of the gut. Same with at least growing hair cells, although they're not mitotic, they're still growing. Another purpose for mitosis is in development, right? So embryonic development. So when sexual reproduction happens, and we get these two haploid cells that have half of the full chromosomes, and that sperm penetrates the egg, we get a new organism, right? We now have a distinct, new, genetically different diploid cell that then begins mitotically dividing and differentiating into and developing into the full organism. So mitosis is used for that too, right? Going from that single cell new zygote to the fully formed organism. Okay, and so eukaryotic cell division, mitosis, happens here, right? So this is the process of eukaryotic cell division. Get my little tool here. Okay, so this is mitosis, right? This whole process, though, is the cell cycle. So if you think about the cell cycle as the lifespan of the cell, mitosis is a part of it. Some cells stop undergoing mitosis, right? So some cells, like brain cells, nervous tissue cells, typically are not mitotic anymore, meaning they're not growing and dividing. And most of your body cells, once you've reached puberty and you stop growing, your muscle cells, your bone cells, they're not mitotically dividing, right? They're just maintaining. But when a cell does go into mitosis, those are the 4 steps, right? Prophase, metaphase, anaphase, and telophase, which involves cytokinesis. This is this guided process of duplicating the chromosomes, right? Duplicating the organelles, dissolving the nuclear membrane, lining up those homologous pairs together, and then pulling those sister chromatids apart into the newly formed cells, right? So we go from one cell like this that's in interphase, it's a fully functioned normal cell, to 2 new daughter cells, right, that are now in interphase. Okay, so the rest of the cell cycle is a process called interphase. And there are kind of 3 key steps in that interphase, particularly for a cell that's going to go into mitosis. G1 is this process of just growing and differentiating. Again, if it's a cell that's not dividing anymore, like a neuron, it's going to be in what we call G0, which is like G1, but it stays that way in perpetuity. That cell kind of survives for the lifespan of that organism. But if it's a mitotic, it's going to be divided and make a new cell. It goes into a process called S phase. S is kind of for synthesis, so that's when the DNA, the chromosomes, right, that hold the DNA, and most of the organelles are going to be duplicated in preparation for making a new cell. And then the last part of that is G2, where the cell is growing in size. It's already been duplicated, and it's getting ready to divide. An interesting thing about this is that there are checkpoints, right? This is a complex path process, irreducibly complex, I would argue. So we've got 3 checkpoints, right? We've got during G1, so G1 to S, and that's going to be a checkpoint, right? So right here, where before the cell progresses to the next step, it's going to go through a series of processes as guided by proteins, cell cycle proteins, checkpoint proteins. Cyclins is what they're called. And these proteins work together to make sure it's safe to move to the next step. And so at this point, in general, it's asking the question and checking the cell: is this cell okay? Is the DNA intact? Is it safe for us to copy the DNA? Right? So then we move on to the next phase. During S phase, so after S phase, it asks: is it safe to progress? Right? Did we duplicate everything okay? Were the chromosomes properly duplicated? Did that process go okay? If so, then we'll move to the next. And then that third key checkpoint would be during M phase of mitosis, right? So at the end of G2, before we move on to metaphase, so right in here, those checkpoint proteins are going to ask, okay, is everything lined up in the middle? Is it safe? Is it ready to divide into 2 daughter cells? Okay, I don't expect you to memorize those cell cycle checkpoints. What I want you to know is the concept, right? The fact that this is a guided process. It's regulated by these checkpoints that are regulated by many proteins called checkpoint proteins, cyclins, and the job of them is to keep damaged, unhealthy cells from being propagated, because that can lead to diseases like cancer. We'll talk about that in a second. And so if these checkpoint proteins work, and they detect an error at any one of these checkpoints, they're going to hit the self-destruct button. They're going to initiate a process called apoptosis, which is kind of programmed controlled cell death, and it basically makes this cell in a controlled way self-destruct and be recognizable by the immune system, so that it can be safely removed and we are not propagating damaged cells. So again, when checkpoint proteins work normally, they hit the self-destruct button and keep damage from progressing. Okay, so again, here's a summary of that. G1 to S is the first checkpoint, G2 to mitosis, and then metaphase to anaphase. Okay, so on the test, I'm going to apply that to a scenario. I want you to be able to think what happens if these checkpoint proteins stop working. Disease in general, but more specifically cancer. Right? Cancer is typically the end product of multiple mutations in multiple checkpoint proteins. So one example is a protein called P53. P53 is a checkpoint protein. Its job is to initiate apoptosis. Right? If the DNA is damaged and it's not safe to replicate that DNA, P53 detects that and initiates apoptosis. In many cancers, particularly many forms of lung cancer, P53 gene, right, so the gene that encodes for that protein, is mutated. And that's a contributing factor to the cancer rate, because this checkpoint doesn't work, and so we can't detect when the DNA is damaged. We miss it, and we propagate that damage, right? And so we get more mutation, which leads to cancer. So again, P53 is a checkpoint protein, particularly in that first checkpoint, right? It works here to stop the replication of cells that have damaged, unsafe DNA. And so that contributes to the process of cancer. So let's talk a little bit about cancer. You've already had the cancer case study, but let me just quickly summarize. So cancer is a mutation or a series of mutations that lead to abnormal, out of control cell growth, and it has to have both of those to be cancer. So simply put, we've got normal cells. So here's a kind of a generalization of tissue layers here with cells. At some point, this normal cell here gets a mutation, right? And there are a lot of things that can cause mutations. Mutations, simply put, right, definitionally, are changes in the DNA, right? Alterations in the normal original DNA sequence. That can happen from just normal growth processes spontaneously, right? DNA polymerase, the enzyme that replicates DNA, can make mistakes. But also mutations can happen through environmental factors, right? We can have a mutagen, a chemical, UV radiation that can contribute to those mutations. At any rate, we get a mutation, and in most cases, it starts just as an abnormal cell. If the abnormal cell just leads to out of control growth, right, just hyperplasia, we would call that a benign tumor, right? So if we've got, this cell's growing out of control, right? And so we can imagine this big ball of cells, but it's not abnormal yet. It's not dysplasia. It's just hyperplasia. That would be a benign tumor. When it becomes cancer is when it has both, right? So in this progression, it's showing a cancerous cell that has multiple mutations. But we get hyperplasia, which is too much cell growth. The cell should not be dividing, but it is. And then at the core of that tumor, you can see highlighted in purple here, we've got abnormal cells. We've got dysplasia. So not only are we replicating mitotically out of control, but we are propagating unhealthy cells that aren't normal, right? They're not behaving like that normal cell, and those are the bad cells, right? So they might make toxins, they might secrete chemicals that enable them to grow blood vessels. So that would be cancer at that point. And then as this cancer progresses, right, to later stages, stage 1, 2, 3, 4, that staging is measured by how harmful that cell is, right? So those abnormal cells, how much damage they can do, how toxic they are to your body, but then also how far spread they are, right? So this process right here is called metastasis, where that tumor breaches its borders. Tiny cancer cells are breaking off. They're going into the bloodstream, they're going into the lymphatic system, and then they're taking root in other parts, right? So that would be later stages of cancer. And we're not going to talk a whole lot about cancer here. You mentioned it in the case study, but basically cancer is hard to treat, because for one, it's small, flies under the radar, but two, cancer isn't an infectious disease, right? It's not a foreign substance. It's you, right? It's your cells that have gone rogue, and our immune system is primed, designed to not attack your cells, right? Otherwise, it would be an autoimmune disorder. So we kind of have this tug of war here where cancer cells can often fly under the radar because, at least at first, they look like a normal one of your cells, and so your immune system doesn't catch it. Sometimes it does, right? And your immune system would fight off that cancer, but oftentimes it doesn't until it's too late, and then it's already spread, it's leaching off other organisms, it's pressing on nerves, and that's when we get kind of the bad news. So then, to kill cancer, we have to either target those abnormal cells, train your immune system to fight it off, which there's some research on that, or a shotgun approach, kill fast-growing cells, right? Which is how typical chemo and radiation works. We're killing fast-growing cells, which are cancer cells, but there are other normal cells that grow fast too, right? Your gut cells, your skin cells, your hair follicles, and so those are some of the consequences of cancer treatment, right? We're killing you without killing you, right? We want to kill your cancer cells, but not your normal cells, and so it's this shotgun approach. Can we kill the cancer fast enough and stop the treatment without going too far and killing too many of your healthy cells? So cancer is quite tough. Okay, then the next layer of this question would be tying in the genetic piece. So the idea here is that some mutations, some cancer diseases are hereditary. What this is a pedigree. So first, let's look at the shapes. Circles represent females, if I can draw here, and squares represent males, right? And so anytime we've got this line here that indicates reproduction, right? These people are married and they had children. So the line that comes down from them and then spreads to other individuals, that's the second generation, right? So the first generation is here, second generation is here, their offspring. And then when we have a new union here, that means one of their children got married and had a third generation, right? So you can see that here. So that's in general how pedigrees work. What they're used for is tracking traits, tracking the flow of genes. So it's often used to track genetic disorders. So if the shape is shaded in, like these, that indicates they have that trait. They have that mutation or that disease, rather. If it's not shaded, it means they don't. Okay. In this more detailed one here, partial would mean they are a carrier, right? They don't have the disease, but they're a carrier for it, so it would be a recessive trait. But we're just going to stick with a simple one on the test. So that's the basics of that. Let's look at an example. So in this example, this is tracking the flow of, let's say, a hereditary genetic disorder. So mom has it here, right? You can see that. I'll do an M here. Well, if I can write. There we go. So this mom has the disease. Dad does not, right? They had two children, a boy and a girl, and they both had the disease. Okay. This girl married this boy who had the disease, and three other children had it. This son had it. He married a wife who didn't have it, and half of their children had it. One of their girls has it. We can use this to kind of figure out the pattern of heredity. So in this one, the fact that it's showing up in every generation, and the fact that the individuals that don't have it are more rare, suggests that it's a dominant pattern, right? So this would be a pattern of dominance, meaning the gene that contains the mutation is passed on in a dominant pattern, meaning all we need is one copy. Remember the way diploid cells work, eukaryotic cells that are adult, they've got two copies for every chromosome, right? Every gene has two copies, two alleles, one from mom, one from dad. That's how sexual reproduction works. And so if a trait is dominant, it means we just need one copy. If we just got one copy of that gene, it's going to show up. We're going to have the trait, right? On the other hand, recessive would mean we can be a carrier, but it takes two of the mutated copies to express, right? We have to be homozygous, recessive, for that trait to show up. And what that would look like in a pedigree would be kind of the opposite. It would skip a generation, right? You might also have two parents who don't have the disease, and they have offspring who have the disease. That's another giveaway that it's a recessive pattern. All right, so that's how we use pedigrees. Hopefully that's helpful. So again, it was an example I would have used in lecture for muscular dystrophy or myotonic dystrophy. All right. We also would use a Punnett square to kind of track the flow of information. Punnett squares, I should use on the other side, but I'll do it here. Punnett squares, right? We draw a grid, and we're just going to deal with a single trait, right? A single allele, a single set of alleles. Okay, and so let's say we'll just use the letter A, right? So big A, little a. So this parent is heterozygous. Hetero meaning they have different copies, one of the dominant allele and one of the recessive. And let's say this parent is also heterozygous, right? So in this case, they are carriers of the recessive trait. They would show the dominant trait, right? They're heterozygous. They've got one copy of the dominant trait, so they're going to have the dominant trait. And so the way the Punnett square works is we just match the letters up in each grid. So this would be big A, big A. This would be big A, little a. This would be big A, little a. And this would be little a, right? So what Punnett squares are used for is kind of estimating the phenotype ratio. So phenotype is the trait, genotype is the combination of alleles they have, the combination of genes they have. And so in this example where we've got two heterozygous individuals, they've got a 25% chance of having a child who has the disease, right? Has the recessive trait. So that's what this shows, that because of the way there are four possibilities of the combination, right? They could both get the dominant and be homozygous dominant. There's a 25% chance of that. They could get one dominant, one recessive. And because they're carriers of both, there's a 50% chance, right? 2 out of 4 that they would be heterozygous. And then there's a 1 out of 4 chance that they would be homozygous recessive. So that's how Punnett squares would work. Okay, so let's erase that and focus on question 3 now. So question 3 will focus on thinking about the flow of information, right? Thinking about molecular biology, connecting it back to some of the things we saw in test 1 with organic macromolecules, how proteins work, how the structure of proteins determines their function. Okay. So the flow of information starts with the genes, right? Everything that makes that individual unique, all the proteins that it needs to make are coded for in the genome, in the DNA. So a gene is a particular sequence of DNA that encodes for a protein or a particular trait, right? So if we need to turn on that trait, we need to make a hormone, right? So we need to make this protein a hormone. The cell's going to get a signal saying, hey, turn on that gene and make that hormone. We need to make that hormone. So once it gets that signal, the first step is a process called transcription, which is what we see here, right? We open up the gene, we make an mRNA, that's what this red molecule is, a message RNA copy or a transcript of the gene, right? So the gene is DNA, we make an RNA copy. It gets processed, so the fully formed product is called message RNA. That's the result of transcription. That's step 1. Once that mRNA is processed, it's put together, it's coded, it's ready to go. Then it's going to leave the nucleus, encounter a ribosome. Ribosomes are made of RNA, and they are enzymes. They're catalytic. They facilitate the production of proteins. These ribosomes might be in the cytoplasm, right, the cytosol, or they might be in the endoplasmic reticulum, depending on whether that needs to be a membrane-bound protein or not. So the second step then is translation, right? So if we coded the message, we transcribed the message, we made a script, right, of the DNA, then we're going to translate that into the product, right? And remember, in our previous one, I talked about this manufacturing economy, right? The DNA are the blueprints housed in the nucleus, which is the engineering department. The mRNA is the copy, right? We need to make that manufactured product. So we send that copy to the manufacturing plant, which would be the endoplasmic reticulum or wherever the ribosomes are. We manufacture the raw product, in this case a protein. That protein might need to be modified, right? So in that case, it would be packaged in a vesicle, a truck, sent to the processing plant, distribution center, the Golgi. It would be modified, right? Have some kind of chemical added to it that would give it its function, perhaps, and perhaps tell it where it needs to go, right? Tell the cell where it needs to go. And so once that processing happens in that Golgi, right, then we package it on a vesicle, which would be the transport truck, right, sending it to its final destination. At any rate, we're looking specifically at these processes. So step one of gene flow, of gene expression, is transcription. Make an mRNA copy of the gene. Step two, take that mRNA copy and translate it, read the code, and make the protein that it codes for, right? So that's the second step. And then we've got the product, right? So in this case, this protein, if it's a hormone that needs to be released, it might be modified, right, in the Golgi, packaged into a secretory vesicle, and then be released into the bloodstream through exocytosis. Okay, so in your reading and your homework this week, you kind of learn how the code works. When DNA is copied, the base pairing rules apply, right? A always pairs with T, G always pairs with C, and that's how you kind of read the code. When we're thinking about gene expression, we have a middle step. We have RNA, right? RNA is similar to DNA, but it's typically single strand, and instead of having T's, it has U's, right? So we'll talk about that flow in a minute, and then of course that would be paired with the anticodon and the protein we made. So there are three types of RNA that we talked about. Message RNA, mRNA, that's the one that's made through transcription, right? That's the copy of the gene. It's going to leave the nucleus and encounter ribosomes, right? So our RNA is ribosomal RNA. It's the RNA that builds up the two subunits of this ribosome, which is essentially this factory that makes proteins. And then the third involved is tRNA, or transfer RNA. There's a tiny bit of RNA connected to the anticodon. The anticodon would just be kind of the mirror image, the pair for the code, right? So every groups of 3 is a codon. So in this mRNA, let me circle it here, right? This would be a codon, AUG. And so the anticodon for that would be UAC, right? It would do the base pairing rule. So U always pairs with A when we're talking RNA. Same here, right? And then C always pairs with G. And so that would be the anticodon that would be here, and it would be attached to the corresponding amino acid, right? That's how the code works. Every 3 base pairs in the gene translates to a corresponding amino acid, and that's how we know to make the right protein. So this mRNA transcript then would be kind of read by the ribosome. The ribosome has a couple of docking sites, the catalytic sites, but the idea is that this tRNA would come in, base pair, codon to anticodon. The next one would come in, the second site, and then the catalytic site here would make a peptide bond between those 2 amino acids, right? And that process would continue until the mRNA transcript is read, the protein is made, and we've made the product, and then it goes on down the line. Okay, so on the test, I want you to be able to think about that process and kind of decode it for me. So let's do a quick scenario. I'm just going to do 2 amino acids in link. So we got let's make up a DNA sequence here. We've got G, T, C, A, T, G. Okay. So if I gave you that 3' here, 3' and 5' just means the direction of the DNA. The enzymes work in a particular direction. Don't get too caught up on that. That's beyond the detail I want you to know. But so let's say I gave you this gene. I say, okay, here's the code for the gene, and it codes for this product. And I asked you if this gene is going to be expressed, we're going to make the product. What's the first step, right? Well, the first step is transcription. So we're going to make an mRNA copy in transcription, right? So then I might ask you to write the mRNA code. So the way you do that, you just use the base pairing rules. So remember, G always pairs with C, A always pairs with T. But since we're going from DNA, I'll do that here, to RNA, right? We're going to make that mRNA molecule. I'll write it underneath here, mRNA. We're going to use U's instead of T's, right? RNA is different from DNA in that way, that it doesn't have uracil, it has thiamine instead. Okay, so let's read the code here, and this would be kind of in the opposite direction, 5'. So this G would be a C, right? So we're going to make this is going to be the first nucleotide in that sequence. All right, and then this would be an A, right? This would be a G. This would be a T, but remember, RNA doesn't have T as a U, so this would be U, and then A, and then C, right? So that would be the mRNA copy, transcript of that gene, right? You with me so far? Then the second step, remember, is translation. That's where we're going to translate it into the protein sequence. That's going to be facilitated by the tRNAs that are bringing in the amino acids. They have the anticodon, and then the ribosomes facilitate that. So the way this chart works, and you will see this chart on the test, is it kind of gives you the code, right? So these will be the so you would read this codon, and it's going to correspond to that amino acid. So follow this with me. The first codon we have here is CAG, right? CAG. So we're going to go over to our chart here, find CAG. Okay, so CAG right here. And GLN, right, which is glycine, would be our first amino acid. So I'm going to write GLN. So that would be the first amino acid in this protein that's being made from this gene. Put a little dash here. And I'm going to go to the next codon, right? So this will be UAC. So let's look at our chart here. UAC, found it right here, will be tyrosine, TYR. And I write that down, right? So on the test, I want you to be able to do that. Again, I'm going to give you a short sequence. It might be a little longer than this one, but that's the process, right? DNA to RNA to protein. So start with the DNA sequence, write down the corresponding mRNA sequence. Remember, we have U's instead of T's. And then we're going to read the code, right? We're going to start with the first three, and then every three of those codons would correspond to an amino acid. We look at our chart, we find where it is, and then we'd write the sequence. Okay. Then the last element of question 3 is what happens when a mutation is involved, right? So a mutation is when we change the DNA sequence, right? We've altered the DNA sequence. There are a couple of different types of mutations we can have. One is a substitution, which means that as the mutation is happening, whether that's happening from the enzyme or some other process, typically the enzyme, we swap the correct nucleotide for the wrong one, right? So we swap an A with a T or a G with an A. So we've just changed one nucleotide and we've substituted it. Substitution mutations may or may not affect the protein. How we know is whether it encodes for a different amino acid, and then whether that amino acid is different enough that the protein would fold up. Remember, that's what drives the shape. We've got the amino acid sequence. Each amino acid has a particular chemical nature. Some are hydrophobic and are going to hide away from water. Some are hydrophilic, they're going to interact with water. Some are positively charged, some are negatively charged, and that's going to drive the folding of this protein into its functional shape. So for this mutation to change that, we have to swap one amino acid for a different one, right? So if we had a positively charged amino acid, and then this mutation now corresponds for a different amino acid, and it's a negatively charged one, that's probably going to change the shape, right? It's probably going to make an altered function protein, probably a defective protein, because, again, if it's different in chemical nature and it's going to fold differently, it's probably going to have a different function. So you can see that here, right? So here's the original DNA sequence, right? CTC. So that's going to correspond to this codon, right? Using base pairing G, A, G. And then we'd look at our chart, and that's glutamic acid. Okay, so that's the normal non-mutated version. In this case, we swapped this middle, this side C with a T. You can see that, right? So that's the mutation. And now that gives us a codon of GAA, which still codes for glutamic acid. So that would be a silent mutation. Same amino acid, same function, no big deal, even though there's a mutation. Another possibility would be that it codes for a similar amino acid. So in this case, we swapped this first C for a G. That gives us CAG, which according to our chart gives us glutamine. Glutamine is similar enough to glutamic acid that it's probably going to fold up the same and not change the function. Another possibility, and this would be the one that changes the function, would be that it encodes for a different amino acid that's not similar, right? So in this case, we've substituted the A for the original T here. That gives us a codon of GUG, which according to our chart gives us a valine. Valine is different in chemical structure, and I would tell you that on the test, whether it's positively charged, negatively charged. But it's different enough that it's probably going to alter how that protein folds and lead to a disrupted function. And then the last step the last possibility would be if the mutation causes translation to stop, right? It added a stop codon. Okay. So then there are a couple other types of mutations that we talked about. A deletion mutation, which would be where we delete a whole base pair sequence, right? An insertion would be where we added an extra one, right? And those will always affect the protein function, right? Because they're going to change the reading frame. They're going to shift the whole sequence and mess the whole thing up. So here's an example. We've got this sentence here, right? The dog saw the cat sit and the fox run. The letters and spacing are precise so that this makes sense, right? It's a sentence. The dog saw the cat sit and the fox run. A deletion mutation would be like taking away this first E, right? So if we did that, and then everything shifts left, this would be the new sentence, right? It's gobbledygook. It doesn't make sense, right? We've altered the whole protein. So insertion and deletion mutations will always change the protein. Substitution mutations, it depends. It depends on which amino acid it substitutes for. Right? So in a test, if I gave you the mutated version and asked you to find it, you should look at the original sequence, look at the new one, and circle where it's different. And then if it's just a single amino acid that's changed, that's a substitution, right? But if you see that the whole thing is shifted right, there's now an extra one in there and the whole thing shifted, that would be an insertion. Or if it's shorter, deletion. And then decode it, right? Go through the same process, look at your chart, find out which amino acids are now coded for. And if it's the same amino acid, the protein function will be unaltered. If it's a similar amino acid, the same thing, probably unaltered. If it's a different and dissimilar amino acid, it will probably change the function. Right? Okay, I hope that helps. These review videos are kind of long, but I hope it's helpful. So just like last time, you can use open notes on your tests. You'll have an hour to take it. Study well. Good luck, and reach out if you have any questions. See you guys next time.